Hello students, in this particular video, today we are going to discuss about cell division under my channel BioWorld. In my first video, I discussed about cell cycle and about the stages of cell division that I would be discussing in this video. Now cell division has to be described fully in the terms of its meaning, that is definition, significance, as well as the stages. Now basically what is cell division? The division word itself means multiplication or growing. Now multiplication of cells for growth or producing new organisms. Now we all know that living organisms need to grow, need to develop throughout their life. So that is first one need of cell division. So cell division entirely is a physiological process in living organisms. So primarily the growth is the first demand under cell division, and continuation of generation, continuation of species is I think utmost demand for the survival and existence of species on the earth. So producing new organisms is also one of the most important demand and requirement under cell division. So basically cell division is multiplication of cells for growth or producing new organisms. It is categorized into two types as I wrote here. First one, mitosis, which is also known as equational division. Now how and why, that I would be telling now. After it we know that mitosis and another one, mitosis is mainly a growth division. So second one is meiosis. Now meiosis is also be known as reductional division. Again, why, that I would be telling you. Now we would start with mitosis first. What is mitosis? The definition, the meaning has to be understood. Like what is mitosis? It's a cell division. We came to know what kind of cell division, what it leads to, why it is called equational division. It's one of the most important question that comes under give reason category, or any sort of question can be given on applicative based or in terms of theoretical question, that why mitosis is called as equational division. So first of all we start with the definition. Student, please do learn this because there are some keywords which I have underlined with the blue color ink pen, that would be easier for you people to understand that these keyword holds marks in the examination. If you don't put this particular word in the definition, you can lose your marks. So what is mitosis? The process by which a cell duplicates, means double, to produce two daughter cells containing same number of chromosomes as present in the mother cell or parent cell. Either of any word you can use. Again, the process by which a cell duplicates to produce two daughter cells containing same number of chromosomes as present in the mother cell or parent cell. So two daughter cells and same number of chromosomes. So the same number of chromosome here, which makes it go to the word equational. Equational means equal. So in this particular division, the parent cell produces the daughter cells, mainly two daughter cells, which has the same number of chromosome as the parent cell. So same, equal makes it equational. So mitosis is known as equational division because of this. The significance of mitosis is growth, repair, regeneration and replacement. That means an organism grow. Every moment it has to grow, there is injury, there is accidental repairing required, or regeneration of any part, or replacement of the part of any part of the physiological system of our body. then a new cell is needed. That time the mitosis plays the key role. So students will continue with the cell division. After the significance of mitosis, we are going to start with the phases of mitosis. So with the diagrammatic representation, which is very much needed for preparing this chapter. So interphase, as we had discussion about interphase in my previous video, let me tell you, in interphase the structure of a cell along with the nucleus and chromatin network is like this, and you have to label them properly in the examination if the diagram comes to you or it is asked to draw in the exam. We need to label it. So without labeling you do not score anything. Remember that. So in a cell, the first thing we need to understand, there is a nucleus, and an interphase, as it is a phase where no division is taking place, so we'll find a simple resting phase where nucleus is there along with a bead-like structure called nucleolus, with a network of thread-like structure which is called chromatin network, as it is not in a condensed form, it is in a network form. And in an animal cell we get to see centrioles surrounded with asters even. We do not find the centrioles in the plant cell, we have to keep it in the mind. And one more thing we need to understand, in plant cell we have a regular cell feature with the cell wall surrounding it as a outer membrane. So leading to interphase. Interphase is the resting phase. After the interphase is completed, the cell gets into active division phase, and a division phase starts with karyokinesis. As I wrote here, you have to understand the meaning of karyokinesis. Now the carrion word has come from nucleus, and kinesis means division. If you can just flash back to your previous classes and get to just get the flashback that what was prokaryotes, what was eukaryotes. Now the carrion word is nucleus. So the same here, the karyokinesis, that is the division that is taking place in the nucleus of a cell. So nucleus divides into two. That is called karyokinesis. After the division of nucleus is over, it is followed by cytokinesis, which means division of cytoplasm. That means when the nucleus has been divided into two, now the term is for the cytoplasm to get constricted in an animal cell with the constriction or the formation of furrow. Whereas in plant cell it is a cell plate, as we know that we have a cell wall which is a very dead and hard covering. So there is a little difference between animal cell and plant cell in terms of cell division. So cytokinesis ultimately divides the parent cell with two different nucleus in the new daughter cells, in the progenies. So that's a completion of a cell division. So we have to undergo the stages of karyokinesis under mitosis. Karyokinesis, that is division of nucleus, starts with the very first stage called prophase. Now prophase is a very important phase in terms of division, and we need to understand with the diagram. Now students, please keep it in mind that as I have not labeled here, you have to label it in the examination, which I am going to tell you how. Because the cell stage, this stage of the cell can come for your examination question, like you have to identify the stage, you have to label them properly, you have to answer the characteristics of the stage during this phase. Now you can see individually plant cell and the animal cell. You can understand by seeing the diagram itself, it's a plant cell, it's an animal cell. So in the prophase, we'll find in the starting phase, initial phase, where you'll find some of the changes happening from the interphase to the prophase, that chromatin network loosens and appears in chromatid fiber. There is a fiber now structure from the network, and there's a point of attachment of the chromatids. Like chromatids are the halves, they are the identical halves of the chromosome. They are called as halves of the chromosome, they are called as hands of the chromosome, and each fiber contains two elongated fibrils remain attached at a point called centromere. This is very important to understand. When we will study about the structure of chromosome also, we will study about it. So we can see in prophase there are number of more characteristics we'll find now. So we'll find the first important characteristics, that is the chromatin here is now condensing and it has come into the form of chromatid, and centromere is the point of attachment of the chromatid. In plant cell we cannot see any centrioles because centrioles are absent in plant cell. But do remember, it is present in animal cell that helps in the formation of spindle during the division of the chromosomes or chromatids. So this centrioles, they have already duplicated and they have moved to the two different sides of the cell. So there are a number of more characteristics. So we found here that after we had a talk about that centriole has duplicated, that again becomes a very important characteristic feature of prophase. The next one I need to tell you that degeneration and disappearance of nucleolus and nuclear membrane is one of the most important characteristic feature of prophase. So gradually we'll find that this nuclear membrane will start disappearing. So this is one of the feature we'll find it, that sometimes they will give you a dotted diagram in place of nuclear membrane. They can give you dotted diagram to understand that nuclear membrane has started disappearing, has started degenerating, so that it has to divide now. It has to dissolve to get divided into two. And next to it, that the centrioles have moved towards the opposite pole, it has already duplicated, and the asters will start forming spindle, and this spindle will get attached like this to the centromere of each chromosome. In the next phase I would be elaborately talking about that how the spindle fibers that has come out from the asters will get to attach itself with the centromere, and then the chromosome will get aligned themselves on a particular plane in the next phase. So summarizing prophase, we found that in prophase already after interphase the chromatin network, the thread-like structure has condensed into chromatid form. This is one of the most important feature that we have come across. And chromatids, they are the identical halves of the chromosomes, and they have been attached with a point of attachment, which we also call it centromere or primary constriction. We have to keep it in mind that primary constriction is the other term for centromere that attaches the halves of the chromosome, that is the chromatids. Gradually the nuclear membrane and nucleolus will get disappeared, centrioles will duplicate and will start moving towards the opposite poles, and aster rays in animal cell will start forming the spindle, and that spindle fiber will get attached to the centromere of the chromosomes. And the same thing we'll find in plant cell, irrespective. One difference is there, that is there is no centriole, and in the place of centriole, the work of centriole is done by the structure called microtubules, that I would be writing it in my next one, and I'll be explaining you that how the spindle fiber formation takes place here. So hopefully children or students, you have liked this video. In my next video I would be more describing about the next stage, that is metaphase, along with the diagrams and the characteristic feature. So please, if you have liked my video, please do like, share and subscribe my channel. Thank you for watching it.